I'll get started. Okay. All right. So my name is Jamil Wilson, and I am a virtual marine science intern at the Smithsonian Marine Ecosystems exhibit. Welcome to our Marine Science in the Morning lecture series. So this series will highlight current marine science research presented by the researchers themselves. And this year we're featuring women in STEM, which means science, technology, engineering, and mathematics. It's an annual winter and spring speaker series at the Smithsonian Marine Ecosystems exhibit at the St. Lucie County Aquarium. So normally this is an in-person program, but this year, for obvious reasons, it's going to be a virtual platform. So information about and links to registration for upcoming programs is available on our Facebook page at facebook.com/SmithsonianSMS. All right, so also, as everybody joins the webinar, I'd like to point out some features. You're more than welcome to use the Q&A box to ask your questions to our guest. The Q&A button is at the bottom of your screen and has 2 speech bubbles. You can submit a question at any time, and we'll try to get to as many as possible before the end of our program. And then also there will be another educator online. She's going to be in the background keeping track of your questions. Erin is her name. So throughout the program, you can also use the chat box to send us messages and answer any questions we may have for you. So your comments are visible only to Smithsonian staff, so please try to keep them appropriate. So yeah, this webinar is going to be recorded and you can find it on Facebook and YouTube, our YouTube channel. All right, so let's get started here. So today on our program, we have Miss Jenny Sneed. All right, so Dr. Jennifer Sneed is a marine microbial chemical ecologist working for the Smithsonian Marine Station in Fort Pierce, Florida, who regularly finds herself swimming through a dark soup of coral gametes. How did she end up here, and more importantly, why? So she'll share her journey and the reasons everyone should be excited about microbes, chemistry, and coral sex. So I'm going to turn it over to Miss Jenny Sneed. And yeah.
Okay, thanks, Jamil. Can everybody hear me?
Yeah.
Okay, good. I'm gonna try to share my screen here. Okay, is that looking good?
Looking good.
See my presentation? Okay, great. Well, good morning, everyone. Thank you for joining us, and thank you, Jamil, for that introduction. As Jamil said, my name is Dr. Jennifer Sneed. I am a research biologist at the Smithsonian Marine Station in Fort Pierce, Florida. For those of you who may not be familiar, we are a research station that is part of the National Museum for Natural History in DC. So we were actually set up to be a place where researchers from the museum could come down, do their research in the field in Florida, do their collections, that sort of thing, and then have support staff there to drive boats for them and help them do their research. So we still function in that way. We have a number of visitors that come down both from within the museum and other institutions, even internationally, come and use our research station. So that's pretty cool. But we also have a number of resident scientists who work year-round at the Marine Station and just do research there. And I fall into that category. So I'm here full-time all of the time. And my work focuses on understanding the chemical compounds that organisms produce that affect the way they interact with other organisms. So there's all kinds of really cool interactions that are going on all around us all the time that we don't really see. I mean, I think probably one of the things that fascinated me the most and brought me into this field the most is the whole idea that trees can even talk to each other through the production of chemical compounds. So it's just, there's a lot of really cool things that are happening in our natural world that we can't see, that are ways of communicating. You may have heard of pheromones that attract mates. Those are chemicals that one organism is producing that attracts another organism. So all kinds of stuff like that. And what I've been focused on mostly lately are things that have to do with coral reefs, and specifically the microbes involved in those. So that's what I'm going to talk to you a little bit about today. But I wanted to start out by saying that you may not realize this, but we are living in a microbial world, and I am a microbial girl. I've wanted to use that title for a long time. But so are you. So I don't know if you know this yet or not, but you actually have more microbial cells in your body than you have human cells. I'll let that sit in for a second. You might be shocked. You're mostly microbial. So what we now know, because we have the technology to look into this, is that human bodies have about 100 trillion microbial cells and only about 30 trillion human cells. So we're really mostly bags of walking microbes. And those microbes are really critical to the functioning of our bodies. So you might have thought, you have this idea in your mind that bacteria are bad for you. That's what causes you to get sick. But we actually have to have certain bacteria in us and on us to really help our bodies function at all in the world and to keep us from getting sick. And there's so many really cool studies that are coming out now that are finding links between the bacteria that live on us and in us and the functioning of our bodies and our health. So I just pulled a few headlines off of Google. I just did a Google search for like human microbiome and health or something like that. And I pulled out a few headlines just to illustrate this. So for example, gut bacteria they're finding may be related to kidney stones, and you may be able to use gut bacteria to treat kidney stones. The bacteria that live in your gut might affect the progression of Alzheimer's disease. So you're starting to talk about like a relationship between your gut and your brain. They're finding that your nasal microbiome, or the bacteria that live in your nose, could be related to asthma. And even our mental health can be affected by our gut microbiome. So they're really finding out now that depression may be in part caused by an imbalance in the bacteria in your gut, which is just crazy to me to think. I mean, if you think about it, you have these bacteria that are living in your intestines and your stomach and whatever, and somehow that's causing you to be depressed if they're not right. I mean, these are really interesting ideas to think about. And so when you think about these bacteria that are functioning in your body, they're doing all these things by producing chemical compounds. So for example, in this study that was looking at the relationship between gut bacteria and mood, what they found was there were a depletion of bacteria in the gut that produced this compound called GABA. And GABA is an anti-inflammatory compound. And so inflammation has been tied to depression for a while, but now we're seeing that it could be related to the not having enough of this compound GABA that's produced by your gut bacteria. And then there was also less bacteria that produce dopamine. And dopamine is one of those neurotransmitters that makes us feel good, right? So it's really interesting to know that there's these bacteria in our gut that are producing compounds that are keeping us happy. And, your mood and depression, I mean, that affects everything in your life. So not only are they affecting, our health and maybe whether we're getting sick or not, but they're affecting our relationships with everyone around us, right? If you're grouchy all the time or you're depressed, that affects your relationship, maybe with your spouse or your significant other. Like, there's all these other things that are being impacted all just by these tiny microscopic organisms that live within us and on us. So I think this is a really fascinating area of science that is really just booming right now. But of course, I don't work on humans. I already told you that. I work on coral reefs mostly. And so here you can see, this is a beautiful picture of a coral reef. You can see this beautiful fish in the foreground. You see these corals, you see some feather duster worms there, maybe some sponges in the background, but you don't see the microbes. And I'm telling you, they're there and they're important. And just like in our human bodies, they are critical to the functioning of these coral reef ecosystems. And in fact, they're pretty much critical to the functioning of any ecosystem on Earth. And so to illustrate this point, I want to tell you a little story about some research that I've been working on for the last 10 years now. I just realized as of February, this is my 10-year anniversary at the Smithsonian. And it starts with these little guys. So I'm showing you a video I took of these little guys that are swimming around in a mixing bowl, actually. Those are about the size of a fleck of pepper. And those little guys will eventually grow into this. So that is the mustard hill coral, Parietes asteroides. And those little swimmers that I showed you before are the larvae of this coral. And so those are basically the youngest life stage of this coral. And then of course, it's corals like these that are necessary to make up these entire reef ecosystems that then support so much life. They support fish life and invertebrate life and have so many critical importances, both ecologically and economically for the coastlines that they surround. So when corals reproduce, I promised to talk about sex. I think I lured maybe many of our audience in by having sex in the title. So now I'm going to get to the sex part of the talk. When corals reproduce, they actually have two modes of reproduction. And so they can either be brooding corals, like this Parietes asteroides, this mustard hill coral I'm showing you here. The mustard hill coral and corals like it, they actually take sperm in from the environment. So sort of more similar to how a human would make a baby, right? Like the mother has the sperm inside and the larva develops inside the coral. And then the coral releases these larvae that you're seeing here. So these are swimming little mustard hill coral larvae. And so when it releases them, the larvae can swim, they're ready to settle, they're all ready to go. The other way that corals can reproduce is what we call spawning. And I'll show you some more examples of that in a minute. But when spawning happens, or corals that spawn, they actually release their eggs and sperm into the water column. So they all just release eggs and sperm all together at one time. Those eggs and sperm swish around in the water. You know, sperm meets egg, fertilization happens. And then over the next few days, the larvae develop in the water column. So they're a little less protected that way. But those are the two main ways of sexual reproduction for corals. But in both cases, the end result is this swimming larval stage, like you see here, these little swimming blobs. Those little blobs will swim down to the bottom eventually, and then they will attach on the bottom and they will metamorphose. So if you see in this picture to the left, there's one, I don't know if you can see my mouse or not, but there's one here on the left that is sort of elongated. That one's still just sort of probing around looking for a good place. And then just beside it, there's one that's already metamorphosed. So this one that looks sort of like a flower, it's already metamorphosed, and that's what it looks like. They sort of flatten down and look like a flower, and then they'll start to grow and develop skeleton after that. And so they go from the larvae to the settled stage, and then with any luck, those little settled baby corals will grow to an adult coral. But it's a pretty tough world out there for a little baby coral, because you can imagine they're just little balls of basically fats, and so they're really delicious to anything that might want to come along to eat them. So we do a lot of our research on this stage, this process of going from this swimming stage to the settled stage, and how the corals decide where they want to attach. So we know that the corals really like this stuff called crustose coralline algae. And so if you see this rock that I'm showing you in this picture that they're settled on, and it looks like it's painted with sort of a pinky purple paint, that's actually an algae growing on the surface of that rock. In the corals, many species of corals really seem to like to attach and metamorphose to these kinds of algae, these CCA, the crustose coralline algae. And so we've really been trying to understand why, like how do these little swimming blobs of fat know what is a crustose coralline algae and what isn't? You know, it's pretty amazing that you see something like this that's swimming around on the left and they can make decisions, but they do make decisions about where to attach. And so that's really fascinating. And we're trying to figure out how they do that. And so, of course, I told you, I spent a long time telling you about all the critical roles of microbes in our bodies. So we thought, could microbes be involved in this process as well? And so to do this work, the first thing we have to do is get the larvae, right? We have to have larvae to be able to do our experiments. So this is the part of the talk where I make you feel really sorry for me for having to go spend weeks at a time on a tiny island off the coast of Belize. You know, it's 14 miles off the coast of Belize. This is our Caribou Cay Marine Field Station. So it's a little lab that's owned by the Smithsonian. And a whole island is only less than an acre big. It's pretty small. It houses 6 scientists at a time, plus 1 cook and a station manager. So that's pretty much all that's there at any given time. And it's a pretty amazing place to get to work because it's also directly on the reef. So it makes it really easy to do some really great research there. And it helps that you have a cook that just makes you all your meals every day, so you don't have to worry about that. So when we go out to try to get larvae from spawners, I told you they release their egg and sperm into the water column. So we actually have to go out and dive after dark because these corals only reproduce once a year. And it's usually within a few days after the full moon in August, depending on the species. Some it's September, maybe October, but it's almost always just 1 month and just a few days of that month that they're going to reproduce. So we have to go out there and start looking at the corals to see if they're going to reproduce so we can capture those eggs and sperm so we can do our work. And so here you can see this is a picture of Zach Foltz. He's our station manager down at Caribou Cay. And he's looking at this elkhorn coral to see if it's going to reproduce. And we can tell if it's going to reproduce because it will put these little balls that contain sperm and eggs. You can actually see some of the balls in the water around him. So some of the other corals are reproducing. But each of these little pink balls has eggs and sperm in it. And they will hold those balls in their polyps of their, in the mouth of their polyps before they release them. And so we go out with a flashlight like you see Zach doing here and checking to see if they're going to spawn. Now my video worked earlier. Here we go. So this is another coral. This is an Orbicella coral, a boulder coral, to show you what spawning looks like. So you can see those pink balls are coming up into the mouth. So you can see it's going to spawn. And then there it releases all of those little pink balls at the same time. And so this is really cool to be out in the ocean. And this is how I said I find myself swimming in a dark soup of coral, egg, and sperm. This is why, because this is what we're doing. We're out there trying to collect these as the spawning is happening, and we have to do it out in the field. And it always happens, at least with these species, after dark. So these little, we call them gamete bundles because each one of those little balls has about has millions of sperm and thousands of eggs in it. And those little balls will actually float up to the surface of the water. And then they break apart at the surface of the water. Here you can see some of the other divers who are swimming through all of that. And it has a very distinctive smell, I can tell you that too. But they'll float up to the surface and they break apart at the surface, and then all that fertilization happens up there. But we can take advantage of the fact that those gamete bundles float And we actually go out and put nets over the corals. So once we see that one's going to reproduce, we put a net over top, and we can collect those little gamete bundles in a cup at the top of that net, because they float up in the net and then they get trapped in the cup, and we just take that cup off. We take it into the lab. This is my colleague, Nicole Fogarty. And we cross-fertilize the eggs and the sperm, and then that zygote starts to grow into a new little larvae. And so this is our wet lab at the Caribou Cay Field Station. So we have to sort of take care of these developing babies for a few days, usually 4 or 5 days, while they grow into larvae. And then we can use them in experiments. So that's sort of the big process of getting larvae to use in experiments from spawning corals, which seems it's pretty complicated. Getting larvae from breeding corals is much easier. So we do most of this work down in Summerlin Key at Mote Marine Lab. For these guys, so this is like, again, this mustard hill coral, we can actually bring them in off the reef, which makes it so much easier. So we can actually go out, we collect these corals, we bring them in, we put them in these mixing bowls. I don't know how many of you have realized that as scientists, we use very specific tools like Rubbermaid mixing bowls, but we actually put them in these mixing bowls, and these guys will release their larvae. Remember, I said they all release fully developed larvae, so we don't have to do any of that fertilization. We don't have to rear them for days before they're ready to use. They just release larvae and we can use them right away. So they'll release their larvae usually right around the full moon, or not the full moon, the new moon in April, May, and June. So we can go out and we take them in off the reef, we put them in this setup, we set it up overnight, and we go out, we have dinner, we have drinks, whatever, we come back, and the next morning we will just have larvae in these containers. So the way we set it up is that we have a water flowing into the mixing bowl. And again, the larvae get released and they float. So then they float across the handle. We've cleverly developed this little water slide for larvae, and they float and they flow across the handle of the mixing bowl into this container. And this container has mesh on the bottom, so the water flows through and all the larvae get trapped. And then we can just use them for experiments. So we like this as a sort of easy setup to do a lot of experiments, because we can get a lot of larvae really fairly easily, So that's how we get the larvae that we use for experiments. And then I started this out saying we were trying to figure out if bacteria are involved in their decisions in settlement. So the first kind of step of figuring that out was to just see if the larvae would settle on tiles that had bacterial biofilms on them. And so we put these tiles out on the reef for various amounts of time to develop a biofilm, which is basically just bacteria growing on the surface of those tiles. So if you put them out for too long, you get other things growing like sponges and tunicates and all that stuff. So we don't want that. We just want the first stage, which is just the bacteria and a few other microorganisms growing on them. So we did that, and we tested this with that mustard hill coral, again, because those larvae are so easy to get. Most of my data I'm going to show you today were done with the mustard hill coral. And so I'm going to show you some graphs like this. On the y-axis, it's always going to be percent settlement or metamorphosis. I think in these graphs, those terms are used interchangeably, but it means that process of attaching and undergoing complete metamorphosis. So sometimes it'll say settlement, sometimes it'll say metamorphosis, but it's the same thing. So if we don't give them anything, which is this FSW treatment down here, this is filter sterilized seawater. So if there's no bacteria there, there's nothing there, they don't really settle. You have just a couple of percent settled. These tiles that were treated for 15 days, you can see we had about 10% settlement. And then the older biofilms had even more settlement. So it seemed like maybe there was an effect of the bacteria, and then we're seeing like once you have a more established biofilm, that the coral larvae like it better. So then we tried to see if we reduced the bacteria in that biofilm by treating with antibiotics, did that reduce settlement? And it in fact did. So you can see this is the natural biofilm, and then that treated with antibiotics here, we had much less settlement. So it really seemed that bacteria were playing some sort of role in causing the larvae to attach to these tiles. So then we wanted to know, is it just any bacteria? Like as long as there's a bacterium there, they will settle, or are there specific bacteria that they are attracted to? And so we went out and isolated bacteria from the surfaces of these crustose coralline algae. You know, I told you they like these CCA, crustose coralline algae. So we isolated bacteria from them to see if there were any specific bacteria we could identify that they were attracted to. And then we did this sort of setup. It's sort of similar to what I showed you before, but this time we have these little discs, these little ceramic discs, and each ceramic disc has only one bacterial strain on it. So instead of being that mixture of whatever grew on the biofilm, these are individual bacterial strains. And you can see in this close-up photo of the Petri dish, you can actually see those larvae. So I told you earlier that they look like flecks of pepper, but I kept showing you magnified pictures of them, but they really do look like tiny little flecks of pepper in there. So when we ran that experiment, we found that out of all the bacterial strains we tested, there was one that induced settlement. So it was this one bacterial strain that we had called PS-MBA5. And we sequenced that, and it turned out to be in this genus Pseudoalteromonas. And so we called it Pseudoalteromonas PS5. And what's really interesting about this bacterial strain is that we isolated this bacterium in Florida from a CCA and showed that it induces settlement in this brooding coral, the mustard hill coral. And it is really closely related to a bacterium that was isolated in Australia that has a very similar function. So they saw that this bacterium in Australia that they isolated from a crustose coralline algae also induce settlement of these different spawning corals. So there's a lot of, there's some crossover here that's happening worldwide with these sort of bacteria that induce coral settlement. Okay, so we now can say, yes, bacteria can signal corals to settle, but whoop, there's supposed to be a question mark there, and there is not. But the next question is how? So how are these bacteria signaling to the corals to settle? And of course, I'm missing a slide here too. I'm sorry. Of course, I told you before that all those bacteria in your gut are producing compounds that affect your brain and all other things. And so it's likely that this is the same thing going on with these bacteria. So what we did was attempted to see if we could isolate a chemical compound from the bacteria that induces settlement. To do that, we grew the bacteria up on marine broth agar. It's just a type of media that we use to grow bacteria, marine bacteria. So we grew it up in culture. We actually just scrape the bacteria off the surface of the plates. We freeze dry it. And then we extract the compounds in organic solvents. So it's just a way to get all the chemical compounds out of those bacterial cells, but leave behind everything else, just the cell walls and all that kind of stuff. And then we added those extracts to sterile seawater in these 6-well plates. So you can see in most of these, there's really nothing else besides seawater. And in a couple of them, you can see little black chunks. Those are actually pieces of CCA that we use as a control to see if the larvae are able to settle on those CCA, because we think they should. They typically like that. So we added extracts just to this water, nothing else. And then we add larvae to those. And we did this with 2 different coral species. We did this with the elkhorn coral, and again with our favorite here, the mustard hill coral. And in both cases, the extracts from the bacteria induced almost the same amount of settlement as just the CCA itself that we already know they like to settle on. So it really seemed promising that these compounds there's some sort of compound being produced by this bacterium that these corals are using to decide to settle where they're settling. And so I just thought I'd take you through kind of how we isolate compounds a little bit. I think probably most people aren't very familiar with this. So when we extract compounds from something like this bacterium, we're getting every compound it has in it well, not every compound, but most of them. And so we can have hundreds to thousands of different compounds in that extract, and it's a whole mixture of different things. So we did the experiment. We know something in that extract is inducing settlement, but we don't yet know what it is. And so to be able to figure out what it is, we do this process called bioassay-guided fractionation. And basically what that means is we take that extract and we divide it into parts. So let's say it had 100 compounds, and we would divide it into 4 groups of 25 compounds each. And then we would test those 4 groups. And let's say one group was active, then we could get rid of the other 75 compounds, right? Like we know it's one of these 25. And then we divide that into like 5 groups of 5. And then we test those and we just narrow it down that way. So one of the first things we do is here what's called column chromatography. This is a way to separate compounds based on polarity. So you put your extracts in here, and there's a solid material in this column, and we put solvents through, and as the solvents move through, the compounds will separate from each other. And so you can kind of see that here in the colored layers that are coming through as the solvent moves through the material. So for this experiment, we did that. We separated our crude extract into 4 parts, and we tested those 4 parts to see if they induced settlement. And so you can see here, basically all the activity was in that first part that we separated, that first fraction. There's a little bit here in fraction 4, but I think that's not, that's just sort of random chance. And then you can see this is again our CCA control that shows we are getting almost as much activity in our fraction 1 here as we're getting in our CCA. So we're pretty confident that whatever our compound is, it's in this first fraction here. So then we had to go ahead and separate that fraction into more parts. So we actually did that. We separated it into 10 parts using what's called high performance liquid chromatography. It's the same exact principle as the column chromatography, except it's done on sort of a finer scale, and it's done through a machine. And so in this machine, there's a detector at the end, and as the compounds come out of the machine, the detector sees those compounds, and you get this printout with a peak for every compound that comes through. And so usually if you have one single peak like this would be hopefully a pure compound, or it might be some very related compounds, but we try to collect those different peaks as they come out, and then we test those. And so we did that, and it turned out our activity was in this peak here, this number, peak number 4. And when we looked at that compound was this tetrabromoperol, is what it's called. And so what we found is that this bacteria that we isolated that induces settlement is producing this compound, tetrabromoperol, that induces settlement in corals. So we were able to identify this chemical compound that's being produced by the bacterium that induces settlement in corals. And we've now been able to test this compound with 6 different coral species from the Caribbean, and it induces settlement in every species that we've tested. So it seems to be pretty widespread. So from here, just going sort of counterclockwise, we have our mustard hill coral that you guys are all pretty familiar with, the mountainous star coral, the this is a brain coral, Copophilia natans. I'm not really great with the common names. I know all the scientific names for these guys. This is the elkhorn coral. This is the pillar coral, and this is a star boulder coral, something, Orbicella franksi. that's its scientific name. But so we know that this compound is pretty widely active amongst, I mean, it's induced settlement in every coral I've tested it with so far. So I think it's pretty commonly able to induce settlement in corals. And so what we're hopefully going to do with that information is be able to help with some coral restoration. So we're really excited to start working with a group called Seacore International. Seacore International is working on doing coral restoration through sexual propagation of corals. Most coral restoration processes to date are based on asexual reproduction. So they do it basically via a process called fragmentation. So they break off pieces of the corals, and then those broken off pieces will grow into a new coral, which is great, but you don't get any genetic diversity using that method. So Seacore is really focused on enhancing genetic diversity by doing restoration through sexual reproduction. And they came to us because they were having an issue with getting the larvae to attach in sort of a uniform way on these tiles that they want them to settle on, so they can put them out on the reef. And so we're working with them to try to figure out if we can use this knowledge that we have now of this compound that induces settlement, or the bacterium that produces it, to somehow attract the larvae to specific surfaces so that they can more easily be basically, so they can be, the larvae can attach to these tiles more easily and more uniformly, and then be able to be put out onto reefs and hopefully grow into new adults. And so we've just started a project working with them on that, and we're pretty excited about it. And, you know, this is really necessary because we have a lot of reefs that are in bad shape right now. And if we want to go from these devastated reefs back to the beautiful reefs of yesteryear, we've got to do some of these cool restoration processes. And microbes are, as I hope you now understand, microbes and chemicals are really important in that process. And I want to end by saying that microbes rule. And I hope I've convinced you of that. So thank you for your attention, and I'm happy to answer questions.
There we go. All right. Can you hear me okay?
Mm-hmm.
All right. So yes, thank you for your time, and thank you for telling us all about coral sex. But yeah, we do have some really good questions, so I'm gonna read them off here really quickly.
Okay.
So one of the questions that we got was, does each pink ball that you guys work with contain sperm and eggs?
Yes. Yeah. So there are different corals reproduce in sort of different ways. There are some corals that are actually male and female. So like some corals have that separation. Some species of corals do, where you know, the males will release sperm and the females will release eggs. But most of the ones we work with have both. And so in each one of those little pink balls, there are both sperm and eggs. And then when they float up to the surface, those pink balls break apart, and then the sperm and eggs get out, and then they mix together and do their thing.
Actually, yeah, that kind of leads to one of the other questions that we had, which was basically how do you guys determine which ones are male or female, or basically, I guess, like hermaphroditic, or
Well,
how do you
so
tell which ones are?
yeah, like
Oh yeah, go ahead.
the ones we work with, we know have both. I mean, there's yeah, the only ones that I've ever worked with in terms of actually collecting the spawn off the reef are that way. So they have both eggs and sperm. And we just, you know, people over the years have studied these different corals, and they know their modes of reproduction. So I've never had to collect just sperm, which seems like it would be very difficult. I don't know. I think they do it with like syringes. I know there are people that do it, and I think they go out and just sort of suck it up in a syringe. But I haven't done that. I haven't collected gametes from those species. So
Dokie. All right. One of our other questions was basically like, what's the rate of coral larvae survival out in the wild?
Yeah, I should know the answer to that,
Yeah,
and I
like
don't really. I'm gonna put that on my list of things to have in my head because I really should know that. But it's not good. It's very, very low, especially for the spawning species. So it's a little higher for, I would say, for the brooders, because they honestly can like put out larvae, and those larvae can just kind of crawl next to the parent and just attach. Like they don't have to go very far because they're already what we call competent. So they're already ready to attach when they're sort of born. Um, but you can think all those eggs and sperm and larvae developing just up in the water, everything's eating them, everything. Like you go out there, when we're out there at night and it's about to spawn, you can tell because the fish are going crazy, the worms are going crazy. It's like everything out on the reef is all excited because they're about to have this smorgasbord of food that's being released from these corals. So yeah, it's very low, the amount that actually survive to grow into a new adult coral.
Gotcha. I have another question for you. I think I might actually know the answer to this, but just for everybody else, I think. So do corals in any way act in the same way humans do in terms of sex and stuff? Like, like I guess releasing gametes for fun or having sex for fun. Is that a thing that's been observed or seen at all?
No, I
Versus
don't
just
think so.
for reproduction purposes?
No, yeah, they just it one of the things that's really amazing about, especially about these spawning corals, is the way they've synchronized to be able to, over large areas, they all release their gametes at the same time because they have to for them to meet in the water column, right? So I don't know, it's just pretty amazing that they can figure it out down to like within 10 minutes of each other. They'll all release at the same time. So it's based on sort of um, things like the lunar cycle, uh, light from the sun, and how soon after sunset, and like there's just all these like intricate factors that work together to let the corals know when to release. But no, I don't think they're doing it for fun at all. I think they're just trying to survive.
Yeah, that's what I figured the answer would be. I was gonna ask just in case, just in case. So during your presentation, you featured some kind of like star-shaped coral restoration objects. Is there a particular reason why they are in that shape? Is it for surface area purposes?
Yeah, so the people at Secor are investigating a number of different shaped tiles, and they are trying to figure out. So you may have noticed there were little grooves in that star-shaped tile, and that's partly because the larvae like to get down in those grooves. So that helps to protect them from getting eaten by like whatever might be coming by to graze or whatever. It's just a little bit of more protection for the larvae if they can get into little nooks and crannies and grooves and stuff. So we know that they kind of like that. And then I think that Secor is also what they're really trying to do is develop a method of doing this whole process that's really sort of easy to do anywhere in the Caribbean. So their goal is that you don't have to have a lab, or like if it's somewhere in Mexico or Belize or somewhere rural where they don't necessarily have facilities, they want to be able to have a way for them to do this kind of coral restoration without a lot of equipment or that kind of stuff. And so what they're trying to develop are these tiles, I think, that you can release like over the reef, and they'll sort of wedge themselves into the reef. So you don't have to have a diver that goes down and puts each little tile in the reef and like places them all, because it's very, that takes a lot of time and a lot of diver effort. So part of the shape thinking is to try to develop shapes that will sort of naturally wedge themselves into the reef and stay there, so they don't just get washed away with a big surge or something. So that's one shape that they've looked at, but I know that they're working on a bunch of other shapes as well. That just happened to be the one they sent us to work with last fall. So.
Oh. So just about one more question for you, which kind of relates. So with the gametes, is it possible to they spawn and stuff at a certain level, or do you have to basically just wait for them to spawn?
I'm sorry, Jamil, you turned into a robot for a second there. I couldn't hear what you said.
Oh, sorry. Basically the question was, is it possible to like freeze the gametes or the larvae so that you can work on them throughout the year and stuff, or do you just have to wait for them to release the gametes?
Yeah, so yeah, we have to be on the coral schedule. So yeah, we have to wait for them to reproduce, which makes it very challenging, especially with these species that only reproduce once a year. So you only have a few days we can usually work with the larvae for maybe a week. Maybe 4 or 5 days before we really can't use them anymore. So yeah, it's tricky because you only have this very short window to do the experiments each year. And then you just hope each year that something doesn't happen. Like one year we were down in Belize and a hurricane came and we had to evacuate the island. And we had all those little developing eggs in those buckets I showed you, and we were trying to rear the larvae, and we had to leave. We had to just hope they survived. We had to leave the island and then came back, I think it was like a day later. And luckily we still had some that were living and we could still do some research, but you just never know. Stuff like that could happen and you have a whole year that's just gone. But that's part of the reason we like to work with that mustard hill coral, because it will reproduce a couple times a year, 2 or 3 times. And so we can get sort of more work done with those guys. And then we sort of, we try to answer a lot of questions with those guys and then test them out with the spawners, because we don't have as much opportunity to work with them.
Right, just kind of extrapolate the info and see if you can make it pervasive across the species.
Yeah.
Okay. All right, well, thank you so much for your time and telling us about, all of the coral sex and stuff and answering all of our questions.
No.
So we really appreciate it.
Yeah.
And thank you everybody in the chat for your questions. Oh,
if anybody's interested, there was, we did have an article in the newsletter recently in the last, so if anybody wants to go to the Smithsonian Marine Station website newsletter, you can see some more information about this work.
Yes, and actually on that note, so for a lecture featuring Ashley Miller, Shelberg from our International Refuge, we'll talk about hatchling oriented turtles one night at a time. So learn more about hatching sea turtles, making their way safely from the beach to the ocean, and hear more from the researchers supporting these turtles' journey. So the registration link should be in the chat below. And thanks for your time. If you are interested in seeing more, please be sure to visit us on our Facebook page at facebook.com/Smithsonian/SMS, or on our YouTube page, youtube.com/SmithsonianSMS as well. All right, and I think that concludes our program for today. So once again, thank you so much, Jennifer.
Oh, thank you. Take care.